Hello, today we're going to be starting session 4, which is structure and function of the brain. And we're going to review organization of the brain and talk about different functional areas of the brain, as well as the electrophysiology differences that we see in these different parts of the brain. So I'm a little biased. My training initially began in neuroscience, studying motor regions of the basal ganglia. But today I'm going to be talking about how that organization of the basal ganglia in terms of motor function is seen in different functions of the brain, including associative circuits and limbic. I briefly discussed this before, but essentially the organization of the brain contains these parallel loops that are partially segregated and process different types of information. However, there is some crosstalk between different parts of the brain. So for instance, when you want to move your arm, there's a motivational area that controls the motivation behind movement that is somewhat communicating with the motor area to initiate motor plans. And as said before, the basic structure is the cortex of the brain, which is the outer surface here, communicates down to some striatal component of the brain, which then communicates to a palatal region, subthalamic nucleus, and then up to a thalamic region, and then back to a reciprocal cortical area. And this structure here, this is the motor circuitry. We can see with associative circuitry, it's a little bit more anterior and goes through the same similar structures, although more anterior in the brain. And then limbic circuitry is a little bit in front of that. But as you can see, the basic structure is the same. And we can see these deep structures, the palatal regions here. The striatal region is in this area. Subthalamic nucleus is down here. This is the thalamus. And then down beneath the subthalamic nucleus is the substantia nigra, or dark body. And substantia nigra means dark body in Latin. As we can see, the structure is carried throughout different functions. Likewise, for instance, in Parkinson's, when you lose dopamine and we administer dopamine to patients to help their Parkinson's state, the dopamine doesn't differentiate between different parts of the striatum. All these different parts of the striatum utilize dopamine. As such, when we try to improve motor function, oftentimes the dopamine can have an adverse effect by altering function, the striatal function of both associative and limbic circuitry, making treatment very difficult and relying on experts with these medications to try to avoid disturbing other functions. The nucleus accumbens is another part of the brain that I studied. The location, this is very anterior in the striatum. This area here is the nucleus accumbens area. I'll be talking about that later on in different sessions, but this area is involved in motivation. And recording in it is different than recording in the putamen section of the striatum, which is in the motor circuitry. Here, this is another area of the brain that's been studied quite extensively now for the treatment of depression, Brodmann's area 25, and we're now using brain stimulation to stimulate what we know now the therapeutic target, which are the fibers above Brodmann's area 25, have therapeutic effect. But we can see that we target different parts of the brain now when neuromodulation treat various disorders. These scans are unique in that it uses an imaging modality which can outline the tracts of the brain, or the axons, which we've been discussing. And it does that in a way by looking at the spins of water molecules. And when water molecules are confined in an axonal space, they orient in a particular direction. So with imaging technology now, we can actually find these axonal fibers and where they project in the brain, from what cortical region to a different cortical region. We can see these long fibers here are going anterior. We have a bunch of crossing fibers in the anterior region. And this simulation just shows the activation zone in those fibers for treatment of this area. Here's another fiber tract. This is the dentate thalamocortical tract. The dentate of the nucleus of the cerebellum is back here. The fibers project up. They cross at midline and then project to the opposite side of the brain. As we've heard, is the left side of the brain controls the right side of the body, and the right side controls the left. However, in the cerebellum, that crossover hasn't occurred. So right side controls right and left side controls left. There are some fibers that remain on the right side. About 10% of the fibers continue up this way, but a majority of the fibers do cross over midline and then up and targeting motor regions. This is a picture of the thalamus. And as we go further into the anatomy, obviously the brain controls our functions. So there's representations of our various functions, and they're encoded in different parts of the brain. The thalamus is in this region here. This particular picture is cutting through the head in this direction, called an axial cut. And what we can see is different parts of the thalamus contain different representations of the motor and sensory region. So for instance, this region here is the encoding of sensory body parts. And we can see that the tongue is very well encoded, and lips and the head. Our hands are disproportionately larger than our back, and our feet are disproportionately larger than our back. And that's because our interface to the environment is highly sensitive to these regions for food seeking, for exploring new objects, for grasping and knowing that we're holding stuff. So the sensory area has an overrepresentation of our interfaces. Up in front of that is the ventral intermediate nucleus of the thalamus. This area receives mostly cerebellar inputs, but we can see the body parts are now much different. The head is smaller. The arms are more proportional, leg and back are more proportional. And this part of the brain actually controls the movement aspects of our behavior. In front of that is another motor region. This is receiving primarily information from the basal ganglia and is involved in movement. Typically, when we're recording these areas, we record mostly involuntary movements in this region and what are called stretch reflexes. So if we squeeze the muscles, we get what are called spindle activations, which are represented in the electrophysiology as a rapid small period firing of the neurons. And we can actually measure the spindle fibers activations in the VIM via the cerebellum. The basal ganglia region mostly responds to volitional movement or the purposeful movement of the patient. So often we'll have the patient move their hand. If we're in the hand region, if we're in the elbow, we'll have them move their arms. What's interesting on this picture is I put a circle representing the diameter of a typical Dixie electrode. And we're going to discuss this more in future slides. But look how a Dixie electrode, when we're recording from the macro contacts, is recording a large volume of the body. We're getting the hand, the back, the buttocks, the thigh, and part of the leg into this recording. So these particular electrodes aren't specific to a body part, but rather will represent the movement of many body parts if you were to record. Here, just a representation. This is the internal capsule, this region here, and we can see the white fibers that come up through the internal capsule actually have representation and orientation of the body. In this case, the tongue is anterior and the feet are posterior. In the thalamus, we can see that the orientation is head and tongue are medial, feet are lateral. And as we're going down through the thalamus, the feet tend to be more on the dorsal section and the head tends to be more in the ventral region of the thalamus. So we can tell along a trajectory where we are in the thalamus by what parts of the body we're recording at the single unit level. And this helps guide our surgical procedures for deep brain stimulation and for various treatments of either Parkinson's or essential tremor, or in the case of pain, we do target the sensory regions. Here's an example on the other side of the internal capsule is the pallidum, and we can see the organization of the body there is much different than both the capsule and the thalamus. And orientation on entry into this region tells us precisely where to place electrodes based on what body parts we see and so forth. Likewise, if we were to stimulate in here, activations of various areas will tell us orientation as well. So it's just not the recordings, the stimulation can be quite helpful in helping orient us in the anatomy. Now, take in mind, this is a representation, and in individuals, the body representations are different. We're all unique. in brain organization. So the thalamus may be smaller in one patient than another. The orientation and position of body parts may be slightly different. So take in mind, we're not identical copies. This orientation map is just a generalized orientation. As I said, the thalamus is broken into many different regions. We have anterior regions more involved in limbic. We have ventral regions, and then we have these other areas that I discussed, the motor and sensory regions. But we can see there's quite a large number of divisions of the thalamus. So when we put an electrode, a macro electrode into this area, we can be in a motor area. At the same time, if we're more anterior, we could be in the ventral anterior as well as the anterior nucleus and a motor region at the same time. So the macro is covering a large, broad space. Micro recordings, however, allow you to have precision in recording individually these structures separately to identify function. And we identify function by having the patients perform various tasks or by the firing rates of the different components. As we see, the subthalamic nucleus has the same type of divisioning. This is the motor region. This is where we try to place electrodes for Parkinson's disease. When we have side effects, it's because our stimulation actually invades these other regions. And that's why we try to avoid placing electrodes near these areas, because we have some adverse effects of various types. So if we go into different parts of the basal ganglia, we see this general organization and separation of body function. We're looking at motor areas again, have separate orientations. And we can see this is the pallidum I described. This is the subthalamic nucleus again. And we can see the orientations are different. However, here is the putamen. This is the striatal compartment of the motor area. And we can see hind limbs are actually anterior and more dorsal regions are posterior. So this organization continues from the cortex through the striatal compartments, through the pallidal compartments, through the subthalamic nucleus, as I've shown, also through the thalamus. However, as we look at this, we had discussed the funneling hypotheses, and that is the scale of and size of these nuclei gets smaller. So for instance, on the cortex, the motor representation exists on the surface of the brain in quite a large region. As that information is encoded down in the striatum, it's now encoded in a smaller region. As we pass through the globus pallidus externus, it becomes even smaller. GPI is smaller. This orientation here, this is off scale, but the subthalamic nucleus is smaller than the pallidum. And then it goes up to the thalamus and then back to the cortex. So from cortex through the basal ganglia, the representations get smaller, areas become closer together, and then they expand back out as we go to the cortex. So the information goes through a funnel back through the cortical regions. So the goal when we do recordings in this region, at least for targeting for deep brain stimulation procedures, is to have very clean signal. And the reason for that is the clean signal allows us to know orientation in the brain, and it allows us to isolate these different body parts as we're projecting through the brain. And that orientation and where we are in the body representation and the fact that maybe the neurons respond to movement tell us exactly where to place our leads. And as we see, just along this trajectory, the thalamus fires in a particular way. The region beneath the thalamus is called zona incerta, or zone of uncertainty. And that's because many different functional areas project through here. And there are some cells, and we can see they fire differently than the thalamus. And then as we insert in to the subthalamic nucleus here, we can see the firing rate is dramatically different than above it. This is a high frequency, very non-regular. However, it does have beta band oscillations. So there are some oscillations, but they fire at a very high rate. In normal brains, we don't see the oscillations that we see in Parkinson's disease, but it's a very characteristic change from zona incerta in the subthalamic nucleus. However, if we project down further out the bottom of the STN and enter into substantia nigra, in particular, this is substantia nigra pars reticulata, we can see that the firing rate is much different in pars reticulata than it is in the subthalamic nucleus. In fact, if we were to template the neurons and count how many spikes are here, it roughly fires at a regularity of about 40 spikes per second. And we can see it's a very, very kind of regular pattern as opposed to this more irregular subthalamic nucleus. So that's the goal, is to have this clean signal to understand where we are orienting in the brain. For research, the clean signal is essential because we're trying to understand how these brains are involved in encoding. In this case, it will be motor function. So how do these different regions encode the ability to move your arm? And having clean signal allows us to see how individual neurons contribute to that function. If we were just to have a macro electrode, we would see how different body parts respond, and it would be a gestalt of all these different activities occurring in that structure, and not a very specific view of how individual neurons are involved in that function. Again, I'm just showing another trajectory. This here is going down into the VIM. So we can see there's some thalamic activity up here. That thalamic activity changes as we enter into the VIM, or ventral intermediate nucleus of the thalamus. It then progresses to a different type of activity, and we know these two types of activity exist in the VIM. And then as we project lower, we enter the fascicularis, which has its own type of activity. We can see these kind of bursting cells down here. This cell is firing at more of an irregular pattern. This one has bursters of low frequency. But this pattern and change allows us to orient within the different regions of the thalamus. This is targeting going into microelectrode recordings into the globus pallidus internus. And we can see as we're entering, we can see distinct changes in activity going through a pallidal region, which is up here. These are the pencils, what we call the pencils of the pallidum. So they're extensions that go up, and we'll enter them, we'll leave them, we'll re-enter them. Then we enter in a GPI, we see a type of activity that's regular bursting kind of activity. This firing rate in GPI is called a pause burst. So it fires, then there's a pause, and then it refires. This is a pause burst neuron. And then in between the GPE and the GPI, we have what's called our border cells, which is a regularity in pattern. We see this, a very distinctive classic border cell. And then we can see we enter GPI, and we have these bursters here. And then we have a different type of pattern, as I discussed as well. So these different trajectories allow us to see different types of activities and help us orient as we're doing trajectories. Again, this is just a reiteration of this funneling hypotheses, which is quite clear here in this picture. The GPI is much larger than the GPE. The pallidum, although not presented, is much larger than the GPE. And then obviously the cortex is much larger than all of these basal ganglia structures. This is just a representation of how the body orientation is on the cortical scale. As we can see, the feet, this picture is a cross section, what we call a coronal section. This line here is at the middle of the brain to orient you. This line here is the middle of the brain. This is the lateral portion of the brain, and we can see the feet are actually in the fold of the brain. The legs extend up through the fold, and then the rest of the body wraps around. This is a very identifiable region. We call this the hand notch. You can see the folds of the brain. We try to identify this hand notch area visibly, and then we can see the head is oriented this way, tongue, and so forth, and different body parts down here. So by recording, we can actually identify these different regions as well. But as I said, this is a general, and in reality, these particular hand regions kind of modulate around over time. So they're not a constant position forever. The brain is a very dynamic and adaptive region. So these body regions kind of shift around. Going back to the thalamus, to reiterate, this is the dimensions roughly of a Dixie macro electrode, so the diameter, and we can see it records many body parts. And as I said, the micro electrode is very small in relation to this, and actually this arrow is pointing to a scaled down dot representing the size and tip of a microelectrode. And we can see how much smaller that is. And in fact, I can place that dot and record a specific region. For instance, I can place it in the hand and record one finger as opposed to the thumb. I can record the forearm area. I can record the biceps. So at the single unit level, I can record function in a much more precise way than I can in the macro level. And I can then ask, how are the hands involved in behavior as opposed to forearms and so forth? And are there differences? Are there similarities? If I'm interested in learning how the body learns how to move your hand for a new behavior, how is that particular hand neuron responding to learning? As opposed to recording this macro activity, which may not be able to see at the precision to tell what the hand's actually doing during a movement. Signal processing. When we're actually looking at these activities, we're looking at the pattern of activities, we're looking at the rate of activities. So for instance, the firing rate up here in the putamen is much slower than what we see down in palatal regions. The firing rates here can go between 20 up to 60 hertz or spikes per second. In the palatal regions, sometimes they fire 2-3 hertz during movement. They may get up to 10-15 hertz, but the sustained baseline activity of the palatum is much higher than the sustained baseline activity in palatal regions. So the firing rate means a lot. It helps us orient to the basal activities of these nuclei. They obviously fire differently during movement. Neurons can either increase their firing rates, decrease. Sometimes they do both, or in a very complicated way during movement. Other things we do is we have software, as I said, and we'll talk about, that help us separate neurons in the different categories. So in this case, we have a microelectrode, and it's positioned between these three schematics of neurons. And these neurons have their own firing rate. We can see this neuron here spiked here, here. This neuron in green spiked at different time points, and the neuron blue fired at different time points. However, we're recording outside the cell. So we're actually recording all this at the same time. When we do these recordings, we rely on software now to help us segregate out the different neurons that are firing. And we do that in different ways. We can look at the amplitude. So this neuron fires about equivalent to the blue and green, but the blue neuron fires when it goes negative, the amplitude is much lower than the red or green. Likewise, the green has a small negative amplitude and the red doesn't have that. So we can sort neurons based on amplitudes. Obviously, they have different shapes. So this neuron, when it fires, has a stereotypic pattern that we see, which is much different than the blue. So we have software that can actually analyze the patterns. So we use amplitude and different morphologies to separate neurons into different categories. And we can see this is an actual tracing, and we can see there's many neurons firing together, and we can see the software has separated these neurons into different groups. Here, the A neuron is found here and here. The C neuron is found here and here. So when I first started in neuroscience, it wasn't too long ago, we didn't have software that was very good at this. The software in recent years has become significantly better at doing this discrimination. In the past, we were recording from 1-2 recordings at a time. Now we have electrodes that record 100 channels at a time. The Dixie micro macro electrodes can record, in one case, 12 recordings at a time or 8 recordings at a time. So manually doing this and reviewing each of these spike trains in the past would take days. Now with our software, we can review and sort neurons in a matter of minutes, half hour to an hour, depending on the length of the data. But as we can see, spike sorting is essential. It allows us to differentiate the individual neurons that are in a recording. So I'm happy to have reviewed all that with you, and I thank you for your attention, and you'll soon be getting session 5.